There are 4 layers of cells making up the retina: a pigmented epithelial layer, the photoreceptors, the rods and cones, several of which synapse with bipolar cells, and several of those synapse with cells of the optic nerve by way of the ganglion layer. In this image, notice how the light travels into the eye and then back toward the photoelectric cells. This seems counterintuitive, but important to know. In this color-enhanced electron micrographic picture, it's clear why the rods and cones are named the way they are. 70% of our body's sensory receptors are found in the eyes and account for 80 to 90% of the brain's sensory activity. The photoreceptors confer information to the nervous system by releasing inhibitor neurotransmitters in the absence of stimulation, that is, when it's dark. When they absorb light, the outer segments split, and in rod cells, for example, causes the light-absorbing pigment called rhodopsin to split into retinol, a vitamin A derivative, and an opsin. This causes a biochemical chain reaction that stops the release of the inhibitory neurotransmitter, allowing transmission of the nerve impulse to the optic nerve. In the cones, the light-absorbing opsin is slightly different. There are 3 different opsins found in the cones, one for each primary light color. Rods are sensitive to low light and detect motion. They do not differentiate color, they see things only in shades of gray. They're found predominantly in the peripheral area of the retina. Actually, they're found all over the retina, but largely around the outside edges. Peripheral vision, therefore, lacks color. The cones contain 3 different opsins, that's light-sensitive pigment, for each color: red, blue, and green. 3 different genes code for each opsin. A genetic mutation can cause colorblindness, a lack of perceptual sensitivity to one of the primary light colors. And cones are concentrated at the fovea centralis, the very center of the retina. There are about 150,000 per square millimeter. This not only enables color definition, but is responsible for high acuity tasks like reading. So, from receptor to neuron, this is an overview of the neurotransmission of a visual stimulus. A photon of light splits rhodopsin into retinol and an opsin. A photon is light energy, and it's enough to split apart the light-sensitive part of the rods and cones. In rods, it's called rhodopsin. In cones, they're called photopsins. The resulting interruption in release of an inhibitory neurotransmission permits a neural impulse to the optic nerve. The biochemical chain reaction causes the cell to hyperpolarize, thus preventing the release of the neurotransmitter. But since the neurotransmitter is an inhibitory neurotransmitter, then its absence causes the nerve impulse to be passed along to the optic nerve. Colorblindness is the lack of perceptual sensitivity to certain colors. The opsins of the photoreceptors are protein. As we will find out in this course, proteins are coded for in our genes. 2 genes on our X chromosome are responsible for the red opsin and the green opsin. The blue opsin is on another chromosome. As such, mutations in these genes confer red-green colorblindness to mostly males. Blue colorblindness is very rare. Absolute colorblindness is unknown. In this picture of the brain, showing the optic nerve transmission pathway, note the optic chiasm, the part of the brain where information from the left field of each eye is integrated to the right side of the brain, and information in the right field is integrated in the left side of the brain.